and the next lesson we will, so you will have to revise it. Where is your calculus? Да, конечно. Focus on stomach, you will revise, yes. And so, and after chyme, yes, after chyme leaves the stomach, then it enters small intestine, yes?
Yes.
So this is stomach, then small intestine starts. So here is, this one is pyloric part of the stomach,
Yes.
and so then it goes to the small intestine. First duodenum.
Then
So small intestine, intestinum tenue, it's in Latin. But it consists of three parts. Duodenum, the first part, and then it continues to the jejunum, and then jejunum, yes, to the ileum. What? No, okay, here there is the ileum. There is another classification, yes? So those parts which have a mesentery, this is a mesentery, like a special ligament that attaches small intestine, no, not only small intestine, like intestine to the posterior abdominal wall, it is a mesentery. So those parts which have mesentery, they are mesenteric part of small intestine, so this is jejunum and ileum, yes? And duodenum is non-mesenteric or fixed part of small intestine. It is also known as mobile because we can see it moves freely. And this one is fixed, yes, like we cannot move it. This is duodenum. Okay, so let's start with duodenum. This is anatomic position. So here we can see liver, then it is stomach, yes, and stomach continues to the duodenum. And so this one is spleen, and this is a pancreas. Pancreas. So this is duodenum, this one. So it is an organ of digestive system, yes, and it performs very many functions. So here in the duodenum, all three types of nutrients are digested, yes. It is due to the pancreatic juice that is excreted here into the duodenum. So proteins are digested by trypsin, and then carbohydrates by amylase, and lipids are digested by lipase. But before lipids are digested by lipase, first they, like large lipids, fats, they get broken down, like they get emulsified by bile, and bile is produced by liver, and bile is also excreted into the duodenum, and also absorption of these nutrients takes place here in the duodenum. So very many functions. About the topography of duodenum, here we can see it. Here it is. This is duodenum, and here this is also duodenum. Yes, it is located mainly in the proper epigastric and umbilical region. And so it starts, its skeletotopy, it starts at the level of the first lumbar vertebra, and it ends at the level of the third lumbar vertebra. And so, but transition of the duodenum to the jejunum, it is located at the level of the second lumbar vertebra. So this is skeletotopy. Then synotopy. So we can see the duodenum is C-shaped organ, yes? Okay, so here we cannot see, but here we can. So duodenum is a C-shaped organ, and it surrounds from all the sides, so from superior, lateral, and inferior sides, it surrounds head of the pancreas. Then laterally from it there is laterally and above there is right lobe of the liver. Behind it there is right kidney, and so proximally there is stomach, yes, and in front of it in front of it there is transverse colon. It is a part of large intestine. So like that. In front of it there is transverse colon. So this is a synotopy of duodenum. Duodenum has four parts. So the first part, after chyme, here by the way, look, here very great preparation. This is pyloric sphincter, yes. So after the chyme leaves pyloric, passes through the pyloric sphincter, it enters the first part, superior part of duodenum, part superior. Then there is the first, like superior duodenal flexure, and superior part after this flexure continues to the descending part. This is descending part of duodenum. Then inferior duodenal flexure at the level of the third lumbar vertebra, yes, continues to the horizontal part of duodenum. It's this one, and then horizontal part continues to the ascending part, yes, from the third lumbar to the second lumbar vertebra, and the ascending part of duodenum at the duodenojejunal flexure continues to the jejunum. So we can see this duodenojejunal flexure. We can see it here. What? So this is ascending part of duodenum. This is jejunum starts. Yes.
Jejunum.
Да, jejunum. Yes. Okay. So these are the parts. Yes. So if we look at the lumen of duodenum, we will see that it has very many transverse circular folds. And so we said, yes, that these numerous folds, we said about it at the previous lesson, that these folds, they are necessary to increase surface area, yes, to improve absorption, digestion, yes, so the more surface area, the more active absorption occurs, and digestion also. And so everywhere there are these circular folds, and in the descending part of duodenum, in the medial surface, yes, there is longitudinal fold. Here it is. Here in the longitudinal fold, there are two papillae. There are two papillae. Yes, this one. This one, the lower one. It is greater papilla. It is Vater's also papilla. So into this papilla, excretory duct of hepatopancreatic ampulla opens. Hepatopancreatic ampulla is formed by junction of common bile duct that comes from the liver and main pancreatic duct which comes from the pancreas. And so they form hepatopancreatic ampulla, and after that it opens in the Vater's papilla. Above it there is lesser papilla. Like it is inconstant. I think it's somewhere here. I think so. But it is inconstant. But it, in most cases, it is present. It is a place of the opening of the excretory duct, of the accessory pancreatic duct. So only pancreatic juice is released through the lesser papilla, and through the greater papilla both bile and pancreatic juice. Yes. So that's what we have to know about the external structure. Now about the internal structure. So as any hollow organ, it has 4 layers. Yes, mucosa, submucosa, muscular, yes, and then the outer layer, what serosa or adventitia. So mucosa is covered by a simple columnar epithelium here, just like in the stomach. Yes, the same. So here there are crypts. No, you will read about it yourself. Okay, and there is also, what is it? I forgot. What is this? Lymphatic. Like this. What is this? Intestinal villi. Yes, there are very many intestinal villi here, and one intestinal villus contains lymphatic vessel as well as blood vessel, because inside the lymphatic vessel mainly lipids are absorbed, yes, and in the blood vessel carbohydrates and proteins are absorbed. Okay, and then submucosa is developed very well because we see how many folds are present in the duodenum, yes, and also in the submucosa there is a nerve plexus. Auerbach, I think, probably I'm confusing. So there are here, one nerve plexus is present in the submucosa, another is present in the muscular layer. One of them is Auerbach plexus, and another one is Meissner's plexus. So also you have to know it. Okay, then muscular layer has 2 layers. Yes, internal one is circular and outer one is longitudinal, and it is smooth, certainly muscle. And okay, and about the outermost layer, actually different parts of duodenum, they have different relation to the peritoneum. So for example, superior part, it is intraperitoneal part, yes, so it is from all the sides covered by serosa, and then descending part, it is mesoperitoneal from 3 sides, and this one horizontal is retroperitoneal, and ascending is again mesoperitoneal, I think. What? No, so in all the sides where there is no serosa, it means there is adventitia here. That was about duodenum. So everything is absorbed here, yes? Once again, I'm telling you, it is trypsin, it is that enzymes are produced in the pancreas, trypsin, and it digests proteins, and then amylase digests carbohydrates, and then lipase, it digests lipids. and bile emulsifies fats, like it splits them into small molecules, and only these small molecules can be digested by lipase after that. So then after all of this, the processes of digestion are over. Then in jejunum and ileum, processes of absorption are very active, and in jejunum they are more active than in the ileum. So there is no clear boundary between jejunum and ileum. We just can say that jejunum, like loops of jejunum, they are located in like left superior parts of the abdominal cavity, and loops of ileum, they are located in lower right inferior parts of the abdominal cavity. So from the outside there is no difference, but there is difference from the inside. So if we look at the mucosa of jejunum and ileum, we will see that they are quite different. Yes. So this is jejunum and this is ileum. First of all, we can see that the closer we are to the large intestine, the less folds are. Yes. First of all, they are less numerous, they are less in number, and also they are less in the height. Yes. So they are not as high as in the jejunum. It's because processes of absorption are the most active in duodenum and then in the jejunum, and by the time when chyme, so chyme, it is, what is it? It is like the food that was treated by gastric and then pancreatic juice, all of these juices. So when chyme reaches jejunum, nearly there is nothing to absorb. Yes, everything has been already absorbed. That's why we don't need such a large surface area in the ileum. One more distinctive feature of the ileum, it is presence of Pierce patches. So what is what are Pierce patches? Here you can see this one and like this one with something rough. Yes, not smooth. This is Pierce patch also. So what is it? These are aggregated lymph nodes. So everywhere in the mucous membrane there are lymph nodes in the whole digestive tract. Yes, because like we need to be protected because food is something foreign. Yes, something not that doesn't have our genotype. Yes, and so but in all the other parts of intestines and in stomach, so everywhere there are just solitary lymph nodes, like they are diffuse, they are solitary, and that's why they are not visible by our eye. They are visible only under the microscope. Yes, and here in the ileum they are aggregated, so they form groups, they are arranged in groups, and that's why they are clearly visible. So this is a distinctive feature of the ileum, Pierce patches. This one, no, yes, you have already seen it. Okay, so here once again I'm telling you absorption takes place mainly. All the nutrients are absorbed and water continues to be absorbed, so everything is absorbed. That was about small intestine. And finally, when from the ileum, this chyme enters large intestine. And so the first part of large intestine, it is a cecum. So this is, here we can see cecum, and here we can see like a boundary in the right inguinal region. There is a boundary, somewhere, let's say, at the level of the iliac crest. Yes, right iliac crest. There is a boundary between ileum and cecum, and here there is a valve. This valve is known as Bohin's valve, or it is also known as ileocecal valve. And here we can also see it. No, like we cannot see the valve itself because we need to dissect intestines in this case. Yes, just I can tell you, it consists of two cusps. Yes, superior and inferior. The function of this Bohin's valve is to prevent backflow of the chyme. Yes, after the chyme entered large intestine, it shouldn't go back. So again, it is unidirectional. Yes, it is Bohin's valve. And okay, then. Everything comes into the large intestine, intestinum crassum, it is in Latin, in Latin, and in Greek it is colon. There are several parts in large intestine, so the first it is cecum, and cecum has, no, here it's not quite clearly visible, it's here, but cecum has like vermiform appendix, yes, this one, This is vermiform appendix. And then after cecum, this one is ascending colon. Ascending colon continues to the transverse colon to the descending. Then it is sigmoid colon, and finally rectum. Here we also can see this is ascending colon, yes. Then transverse colon, then it is descending colon, sigmoid colon, and rectum. Yes, here we can see it. Okay, so if we are talking about the topography, yes, we see that loops of small coils of small intestine, they are located in the center, yes, and large intestine is located in the periphery. Okay, we also have to know how to distinguish large intestine from small intestine. Large intestine from small intestine. This is small, and this is large. So what is the difference? First of all, we can see that small intestine is like clean, smooth, yes? Nothing is attached to it, yes? There is no fatty tissue that is attached to it, so, and certainly, if we compare diameter, certainly small is smaller and large is larger, yes? It is obviously, okay. And to this large intestine, different, like, appendages are attached. In the transverse colon, this one is greater omentum. And in the other parts of large intestine, these are just fatty appendages here, everywhere, just different fatty tissue derivatives, yes? So the other sign, the other distinctive feature of small intestine is that it is smooth, yes, as I told you. And here in the large intestine, longitudinal layer of muscular tissue forms three tenia or ribbon-like structures, yes? So all these tenia, they originate from the appendix. So at the beginning, at the region of the appendix. And so if we start from the appendix, we will see that this one is tenia libera, free tenia, so nothing is attached to it, yes? Then another one, this one, it is tenia mesocolica. No, here everything is broken, but here we can see it very well. This one again, this one is tenia libera, yes. This one is tenia mesocolica, where mesocolon, again this ligament, yes. What I forgot to say, that in Greek, small intestine is enteros. Da, enteros. And here it is colon. That's why here this ligament is named mesentery, and here it is named mesocolon. What? So mesocolic tenia, it is a place where mesocolon is attached. And the third tenia, it is omental tenia. For the transverse colon, omentum is attached to the omental tenia. And for the other parts of colon, these are just fatty appendages are attached. No, this is like, he is very thin. Let's see. It's not, this fatty tissue is not well developed, but here it is quite well. Look. So this one is tenia libera, free tenia, and this one is tenia mesocolica, yes, where mesocolon is attached, and this one is omental tenia, where omentum is attached. So here we can suggest that it was transverse colon, yes. One more distinctive feature of large intestine, it is presence of hostra. These are like how should we say, like sacs, yes, or like elevation protrusions, let's say so, which are between this tenia, No, here in the small intestine again we could not see them. Here, let's take a look. This one is hostra, yes, this one, like between tenia they are located. If we compare large and small intestine in a live person, we will see that color is also different. So for the small intestine it will be pink, and for the large intestine it will be gray. It's due to the location of vessels in the wall of the intestine. So in the small intestine, these vessels are located more superficially, and they are visible in the wall, yes. And in the large intestine, they are deeper. That's why large intestine has a gray color. Fine. So what are the functions? Ah, yes, wait. And certainly from the mucosa side, it is also different, yes? So if we compare small intestine with large intestine, we'll see that there is even less folds than in the jejunum or than in the ileum, yes? And they are not circular, they are semilunar, and they are very, like, no, they are very few. Because, yes, what are the functions? Due to the functions of large intestine. So absorption of nutrients is over in small intestine. So here water is still absorbed and salts are absorbed and some vitamins are also absorbed here in large intestine. And another function due to this absorption of water and salts, feces is formed. So formation of feces is another function of large intestine. Yes? Okay. And so that's why folds are not so high. So it means if the folds are not so high, it means submucosa is not so well developed, yes, here, if to compare it with the small intestine. Okay, so as I told you, there are several parts, yes? You have to know the topography. So a cecum starts at the level of the right iliac crest and it goes to the false pelvis, yes, it's dome, it's like, what is it? No, like inferior part is directed to the false pelvis. And so at the level of the right iliac crest, cecum continues to the ascending colon, ascending because it ascends, yes, goes upward. Then here there is, below the right lobe of liver, yes, there is right colic flexure or hepatic flexure. So here, ascending colon continues to the transverse colon. And then at the left colic flexure, at the level of spleen, it is also known as splenic flexure. Transverse colon continues to the descending colon. This one is descending. Then at the level of the left iliac crest, descending colon continues to the sigmoid colon. And then at the level of the promontory, sigmoid colon continues to the rectum. Yes. No, actually, structure of all of this. Okay, we didn't discuss it, yes? Okay, again, it is hollow organ, and again, there is mucosa, and it is covered by simple columnar epithelium, yes, and then some mucosa that is not quite well developed, and then there is muscular layer, the like internal layer is circular, and then external one is longitudinal, yes, and then from outside, for the transverse colon, there's serosa from all the sides, and for the sigmoid colon as well. And for example, ascending and descending colon, they are mesoperitoneal, so it means from three sides they are covered by peritoneum. Peritoneum we'll study at the next lesson. Now you will have just to memorize it, but at the next lesson we will discuss it in details. Okay, and so then finally everything goes to the rectum. Так, where is rectum? So this is rectum. Rectum, in Latin it is also rectum, and in Greek it is proctus, yes? So that's it. This is internal side, this is external side, this is anus, yes, because here we can see skin. So the function of rectum is, Здрасте. Sure. We are about to finish. So function of rectum, it is storage of feces and excretion of feces, yes? So this is rectum, I told you, and yes, topography. It is located in the true pelvis, and so please revise what is actually true pelvis, yes? So its superior boundary, if we are talking about skeletotopy, is at the level of the promontory, so this is rectum, yes? At the level of the promontory it starts, and then so it goes downward to the perineum, and there it ends. Okay, syntopy, yes, very interesting. So syntopy of rectum is different in males and females. In males, in front of the rectum, there is urinary bladder, yes? And in females, in front of the rectum, there is uterus, yes? And only after that, urinary bladder. So behind there is sacrum, and above there are loops, coils of small intestine, and that is all. Okay, so about the external structure, what we can see? There are parts. So this dilated part, it is ampulla. And after that, it is anal canal. I think in your books there is something like preampular part also. Okay, you will read it. So this is ampulla, dilated part, and then it continues to the anal canal. Continue. Ampulla, it is dilated part where feces first stored, yes, and at the time when it is like much enough to be excreted, then it goes to the anal canal. This is anal canal. So in the anal canal there are anal columns, columna analis, here they are, yes, and between them there are like anal sulci, sulci analis. Here there are special mucous glands, yes, which produce mucus, and that is all. And so yes, we can see that in the anal canal folds are oriented longitudinally, and in the ampulla folds are oriented circularly, yes, transversely, yes. Okay. Okay. So every day, no, usually in normal cases, yes, 150 milliliters of feces is stored, no, is produced and then excreted. Yes, so here 150 milliliters should be should stay. Okay, now about internal structure. Yes, so there is mucosa, and it is covered by, in superior parts it is covered by stratified squamous non-keratinized epithelium, and at the region of anus it is keratinized, yes. And then there is some mucosa, no, it's not so well developed, but still it is present. And then there is a muscular layer, and the internal layer circular and external is longitudinal as always, yes. And here we remember, yes, that here there is another transition from the smooth to the striated muscular tissue. So there are two sphincters. At the level of anus, there is external anal sphincter. Okay, external anal sphincter, that is voluntary, yes, that is formed by striated muscular tissue. And above it, there is internal anal sphincter, that is involuntary, that is formed by smooth muscular tissue. So that is what. Yes, and we can also conditionally divide rectum into three parts when we describe the outer layer. So in the distal part, it is totally covered by adventitia. Then here in the middle part, it is covered from one side, so it is retroperitoneal, it is covered by serosa from one side. And in the upper part, it is mesoperitoneal, so it is covered by serosa from three sides. And from posterior side, it is covered by adventitia. That is about rectum. So that was a digestive tract. What questions? No questions.
No, how many valves in the digestive system?
How many again?
Valves.
Valves.
Yes, like construction.
Very interesting question. I have never counted.
Okay, but you told us there is one of the pyloric.
Yes, there is.
Yes, in the pyloric, and
yes,
there is another one in the intestine.
There are more than one. Usually, the sphincters are located in, but for example, yes, in the transition between different parts, like between ileum and large intestine and cecum. Okay. Yes, so yes, they are present.
Valves, it's the same meaning as sphincter?
Yes, no. Valves, it is like they are formed by, it's like a barrier, just like a door. And sphincters, it's like thickening of circular layer of muscles. So valves are not muscular structures. Okay, one more thing, not one more. So, more than one. But we also have to know about parenchymal organs, about digestive glands. So one of them is liver. This is liver, and this is also liver, yes, and this is also liver, yes. Heparit is in Greek, so it is digestive gland, it is organ of digestive system, but actually it performs many other functions, not only regarding digestion. So yes, regarding digestion, it produces bile, yes. Bile serves for emulsification of fats, but also it performs function of detoxification and production of clotting factors and storage of glycogen. So glycogen, it is like large molecule that is, like it is polysaccharide, let's say so, like glucose. Many glucose molecules, they join together from glycogen, and it is stored in liver and in muscles until it is required. So very many functions. And in the fetal period, liver also performed function of hemopoiesis, production of blood cells. Okay, that's what you have to know. So about the topography, no, skeletotopy you will read. yourself. Then about holotopy, it is located in mainly the right hypochondric and proper-epigastric region, but even some part goes to the left hypochondric region. And so syntopy also nothing is complicated, yes. So above there is a diaphragm, and then right lung and heart, and some part of left lung even. And so to the left from the liver there is a stomach, and below it there is transverse colon, and also there is gallbladder. Gallbladder. You also have to study gallbladder. I forgot to say about it to the other group. I will tell them. Так, yes. And so below it also there is right kidney. Right kidney. Not so, yes, syntopy is simple, you will also study it. So this is again anatomic position, yes? So in the liver there are two surfaces. Diaphragmatic surface is that one that is directed to the diaphragm. Yes, this is diaphragmatic surface. And then visceral surface is that one that is directed to the other internal organs. They are connected with each other by two borders. So this one is anterior border or inferior border, it is sharp. And this one is posterior border, this is smooth. This is posterior border. In the liver, mainly in the diaphragmatic, let's say so. In the diaphragmatic lobe of surface of the liver, two lobes are distinguished. Larger one is right, lobus dexter, and smaller one is left, lobus sinister, yes? So they are separated from each other by falciform ligament, ligamento falciformis. It is also formed by peritoneum. We will talk about it later as the next lesson. Okay, in the visceral surface there are two more lobes. Next time we will have to take another liver.
Which
Yes.
has lobes? Which the lobes is separated?
Yes, we'll see. So there are two more lobes. So there is right and left again, and there is also quadrate and caudate. So actually they do not look like this. It's the compressed liver. Okay, so you have to know the boundaries. And also, what we forgot to say, that in the visceral surface of the liver, in the center there is porta hepatis, like the gates, the entrance into the liver, through which vessels, nerves, bile ducts, they enter and leave. Now this is very good preparation. So what can we see here? Так. So this one is bile duct, yes, common bile duct, and this one is portal vein, and this one is hepatic artery. This one, hepatic artery. So these structures they enter the liver. So what is interesting? This one duct, and this one let me think. This one vein, and this is artery. Yes, I think this is artery. Looks like artery. Okay, so what is interesting? That liver is a unique organ, no, except the heart, if we don't count the heart. Okay, it is a unique organ, the only organ into which both arteries and veins enter. So hepatic artery enters it, and portal vein also enters. Usually veins leave only, yes, and here both portal vein and hepatic artery enters, and moreover, portal vein actually brings 80% of blood to the liver, and hepatic artery gives only 20%. So portal vein brings venous blood, 80%. Portal vein brings 80% of blood, and hepatic artery gives only 20%. So, why does portal vein enter liver? Because one of the functions of the liver is detoxification, and portal vein collects venous blood from organs of digestive tract, from the intestines, and so it contains very many nutrients, and also it contains probably some toxins, and they should be detoxified by liver first, and only after that to go to the heart and then to the other organs of our body. Okay, and so what else can we see on the visceral surface? We can see here, this is gallbladder, so we can see gallbladder fossa here, and it is from the right side. Yes, so this one is gallbladder fossa from the right side, and from the left side we can see this one is teres ligament, round ligament. So here there is groove for round ligament. No, it is here. And then posteriorly from the groove for the round ligament, there is groove for the venous ligament. And posteriorly from gallbladder fossa, there is fissure for inferior vena cava. So this one is inferior vena cava. So now we have to know the boundaries of quadrate and caudate lobes. This is quadrate lobe, lobus quadratus. This is caudate lobe, lobus caudatus. Cauda means tail. Yes? Done. Okay. So boundaries of quadrate lobe from the right side, from the lateral side, it is groove for the, gallbladder fossa. Then from the left side, it is groove for the round ligament, and posteriorly it is porta hepatis. For the caudate lobe, the boundaries are anteriorly it is porta hepatis, and then from the left it is groove for the venous ligament, and from the right it is fissure for inferior vena cava. So these are the boundaries of quadrate and caudate lobe. And that's about internal structure. Now certainly you have to know that in the visceral surface there are different impressions, like this one is impression for the stomach, here there is impression for right kidney. You will read it in the book, everything is very simple. Yes. Okay, that was about the liver, external structure. About internal structure of liver we will talk a little bit later. And one more thing that we have to study for the next lesson, that will be gallbladder. You will read it, very simple also. Yes. And pancreas. Yes. So this is pancreas. This is pancreas. This is pancreas. Here is pancreas also. What this one? Head and tail, or body, and tail, pancreas. And let's take a look here also. So this is like, they are covered by peritoneum, but anyway it is pancreas, head and body, and tail. Tail goes up to the spleen. So pancreas. So liver was exocrine gland. Yes, exocrine gland means that gland which produces enzymes, some substances, and it excretes its substances onto the surface of skin, like sweat glands, or into special another hollow organs, into canals. And pancreas is a gland of mixed secretion. So it is both exocrine and endocrine. As an exocrine gland, it produces pancreatic juice, and this juice finally is excreted to the duodenum. Yes, we have already said it. And as endocrine gland, it produces different hormones such as insulin and glucagon and some others. Now we are interested only in these two. So these two hormones, all the hormones, are excreted into the blood, and these two hormones, they regulate level of sugar, level of glucose in the blood. Now, so about the topography, we didn't say, yes, topography. So it is located at the level of the second lumbar vertebra, yes, second lumbar vertebra, and tail, I think, goes even to the first, yes, lumbar vertebra. Okay, and it is located in the umbilical and left lateral region. And about synopy, we can say that head of the pancreas is surrounded by, here we can see it, by the duodenum. Yes, head of the pancreas is surrounded by duodenum. Above the pancreas there is stomach, and laterally from the pancreas there is spleen. And below the pancreas, and first of all, let's say in front of the pancreas there is transverse colon. And yes, and behind there is left kidney. Behind the tail and body of pancreas there is left kidney, and so on. About the external structure of pancreas, it has head, here, a caput, then body, corpus, then tail. It is like trihedral. It has a trihedral shape. It has inferior surface, anterosuperior and posterosuperior, and it has three borders: superior border, anterior inferior, and posterior inferior border. And that is all about the external. No, not all. Between head of the body of pancreas there is a notch. Notch. I don't remember its name, but you will read it in the book also. Okay, done. Yes, if it is parenchymus organ, and it is parenchymus organ, you have to know what is morphofunctional unit. Yes, what is the smallest part of the organ that can perform all the functions of it. So because it is both exocrine and endocrine gland, so as it has different morphofunctional units for exocrine and for endocrine part. For exocrine it is pancreatic acinus, and for endocrine gland it is endocrine part it is islet of Langerhans. Islet of Langerhans. And so you will read about it. For pancreas you just need to know what are the names. Yes, there is one more organ which you also have to study for the next lesson. It is spleen. Here it is, but you will read it yourself. So now, please, any questions? No question. So now, please take your seats. So morphofunctional unit of liver is hepatic lobule. Liver performs very many functions, yes? And so that's why only one cell cannot perform all of them. That's why cells are arranged into lobules, and this lobule can perform all the functions of the liver. So lobules are polygonal, usually hexagonal, yes, like 6 sides, yes. And in the center of hepatic lobule, there is central vein. And away from the central vein, rows of hepatocytes arise. So rows of hepatocytes. Site means cell, hepato means liver, so it is just typical liver cell. This is the nucleus. What? 2 rows of hepatocytes, rows. Do you understand the word row is? Row.
Yes.
2 rows of hepatocytes, they form hepatic cord. Hepatic cord. Hepatic cord. This one is hepatic cord, and this one is hepatic cord, and this one is also another hepatic cord. So between 2 hepatic cords, there is sinusoid. So sinusoid, it is capillary, and it has wall, and wall of the sinusoid is formed by, as any capillary, yes, it is endothelial cell, but here it is like in normal capillary, usually there is basement membrane, yes, under the endothelial cells. Here there is no basement membrane, and that's why permeability of the sinusoid is very high. It means substances which are now in the blood of sinusoid, they can easily leave bloodstream and go to be absorbed by hepatocytes, for example, yes? Because there are gaps, very big gaps between endothelial cells. So this is sinusoid. Sinusoid contains mixed blood. Because as I told you, into the liver both portal vein and hepatic artery enters, yes? And so that's why blood from hepatic artery and from portal vein, it gets mixed, yes, and it enters sinusoid. And blood flows towards the central vein, in the direction to the central vein. While it flows towards the central vein, it is treated, yes? How? So here between endothelial cells and hepatocytes, there is a space. It is space of descent. Here in this space there are phagocytes. These are not macrophages, or they have special names. These are Kupffer cells. Yes, Kupffer cells. And so they intake all the toxins. So they destroy them. Yes. So toxins are destroyed by Kupffer cells. And nutrients like glucose, for example, all the nutrients, they are absorbed by hepatocytes. And so glucose is, for example, stored. Glycogen is stored here, and some nutrients are used to produce bile. And also oxygen is taken, of course. And that's why this mixed blood, when it finally reaches central vein, becomes venous, yes, deoxygenated in the central vein. So here there is sinusoid, and here there is also another sinusoid, and here there is sinusoid also. And inside the hepatic cord, between 2 rows of hepatocytes, there is bile canaliculus. This is. And this is also this. Let it be. Bile canaliculus. Bile canaliculus. So hepatocytes, after they receive nutrients, they produce bile. And this bile is excreted to the bile canaliculus. And bile flows outward from the central vein. So blood flows towards the central vein and bile outward. And the blood like that, yes? Like that. And that. Okay. And so that's how it works. And actually we know there are very many hepatic globules, yes? Very many hepatic globules. And here there is hepatic globule. And here there is another one hepatic globule. And so everywhere. And so at the place of junction of hepatic globules, there is hepatic triad. Actually, it's somewhere written hepatic tetrade. Well, okay, it's also correct. Just for now, we're interested only in three structures. So this hepatic triad includes three structures. It is interlobular, artery, and interlobular vein, No, so the portal vein and hepatic artery enter the liver. They form many branches. They are smaller and smaller, and finally they form this interlobular arteries and veins. Inter means between. Yes? Yes. Lobule means lobule, so between lobules. And so blood from interlobular artery and from interlobular vein, it goes to the sinusoid, and so it mixes and forms mixed blood of sinusoid. And the bile from bile canaliculus goes to the this one, the third structure, is known as interlobular bile duct. And so bile from bile canaliculus of all the adjacent lobules, it goes here to the interlobular bile duct. That's how hepatic globule functions. This diagram you will have to draw, all of you, at the beginning of the next lesson.
It's present on the book.
No, some variations are present.
مش زي كده اصلا موجوده في احنا مش الشكل
صدقني مش هتلاقي احسن من الفيديو ده خلاص
Okay, any questions? No questions. So you read, like you revise, yes, esophagus, stomach, and then you read digestive tract up to the end, yes, and then you read liver and pancreas, spleen also everything, yes.